Hi, today I'll be discussing how we can achieve accurate population estimates of the elusive snow leopard. Population size estimates of snow leopards are used for making important conservation decisions, so getting accurate estimates is imperative for the future of this threatened species. The problem is that it is very difficult to attain these accurate estimates. For large carnivores in general, it is difficult as they occur in low densities, have wide ranges, and are cryptic. For snow leopards in particular, their habitat is largely inaccessible due to the difficult terrain and high altitudes. Our current sampling methods are inaccurate and biased, and most current studies have failed to produce robust estimates. There are currently two methods that are recognized as the most accurate. I will analyze both camera traps and fecal DNA estimation methods and state possible modifications that can improve estimation accuracy. The first method I'm going to evaluate is camera trapping, in which remote sensor field cameras are placed across the study area. Study areas are typically chosen for places that are preferred and frequently traveled by snow leopards. Individual snow leopards are identified using spot patterns, and then these individual capture histories are used to estimate abundance and corrected for imperfect detection. The pros of using cameras is that they are low cost and can remain in the field for long periods of time. They make sampling in the rough terrain easier, and population estimates are typically robust if there are enough captures. Cons associated with using this technique is that individual identification can be difficult, for example if the photos are unclear and blurry or spots are distorted. Another big assumption here is that spot patterns can even be used to identify individuals, as this has not been tested. It can also be difficult to get all of the necessary captures for robust estimates, and of course technical malfunctions may occur. There are a number of possible modifications that can be made to improve this technique, such as using scent lures and camera trap arrays to increase detection, although each of those must be corrected for potential bias. The ability to ID snow leopards based on spots should also be tested. Estimates attained can be standardized with ungulate prey abundance surveys or sign transects. Future technologies, such as facial recognition software, may be useful for faster individual identification. The next method is fecal DNA sampling. For this, samples of snow leopard scat are collected from a study area. Material is taken back to a lab and individuals are identified by extracting genetic information from the samples and analyzing the DNA. Individuals identified are used in capture-recapture modeling to estimate abundance. The pros of this method are that accurate individual identification can be done even with small amounts of material, and the dry environment of the snow leopard keeps scats well preserved, so that makes collecting even easier. Snow leopards also have predictable marking sites, meaning sampling can be more efficient and large-scale surveys can be done at low cost. Estimates can be very accurate, and DNA can give us other information, such as diet, as shown in the pie chart. Problems occur when the DNA is poor quality, as identification can be difficult. If scat is incorrectly identified in the field, resources are wasted on sequencing, as shown in the chart where more than 50% of most samples taken were not snow leopard. There may also be violations of some modeling assumptions, as subadults could be included in the samples, and scat lasts a long time in the field, so the population may not be closed. Possible modifications for this method include sampling from larger areas to prevent positive bias, and placing sample transects in a variety of locations. There are also some modeling considerations, such as making scat depositions a factor to correct estimates, and using variable density to correct if the sample area is too small. You can also use hierarchical spatial capture-recapture modeling to account for the imperfect distribution and detection of scats. No one method is perfect for estimations, especially with the difficult terrain and elusive nature of snow leopards. My suggestion is a combination of the methods to boost detection probabilities and improve accuracy. However, if money and time constrains the study to one method, as is often the case, then I suggest fecal DNA sampling with the modifications mentioned previously. Field effort, total time, and cost is less compared to camera trapping, as is shown in the table. On larger geographic scales, DNA methods have higher detection probabilities than cameras. The nature of snow leopard ecology and their habitat makes DNA sampling easier, and DNA also gives us other important information about the cats, as shown in the graphic. A combination of methods would be ideal, but given study constraints, fecal DNA sampling provides the best and most accurate information. Here are my sources, including the papers I used and the picture credits in order of appearance. Thank you all for watching, and please feel free to ask questions and leave comments below the video.